The ability to insert or knock in a gene at a specific locus is a tremendously useful method for dissecting disease mechanisms. However, in previous years, the insertion of a foreign gene has been far too tedious and expensive for many labs to perform. Thanks to the recent development of CRISPR-Cas9 technology, gene knock-in experiments have now become a very popular technique in modern molecular biology. In contrast to the random integration events by the lentiviral system, CRISPR utilizes a guide RNA that directs gene insertion with extraordinary specificity. In particular, the AAV-S1 safe harbor site is often a preferred target site for gene knock-ins. This is because it enables consistent and robust transgene expression with minimal toxic effects. At this safe harbor site, the surrounding DNA maintains an open conformation, enabling stable expression of the newly inserted transgene. This makes safe harbor sites, such as the AAV-S1 locus on human chromosome 19 and the ROSA26 locus on mouse chromosome 6, very popular sites for targeted knock-in experiments. In this case study, we will walk you through how a red fluorescent protein gene was knocked into human embryonic kidney cells at the AAV-S1 safe harbor site. We will also demonstrate how the knock-in was verified using a PCR-based method. To begin, an sgRNA was designed to target the human AAV-S1 locus. Software analysis was performed to ensure maximum efficacy of on-target cleavage while avoiding any off-target effects. The resulting guide sequence was used to construct ABM's AAV-S1 targeting all-in-one vector. This vector expresses the Cas9 endonuclease under a CMV promoter and the sgRNA under a U6 promoter. To achieve the knock-in, a donor plasmid containing the gene of interest is required to provide a template for homology-directed repair. The HDR pathway is a mechanism in cells that repairs double-stranded breaks by using a DNA repair template homologous to the sites flanking upstream and downstream of the break site. To enable HDR-mediated gene insertion, the gene of interest must be flanked by such homology arms. Therefore, a second plasmid was constructed by cloning an RFP pyromycin expression cassette into ABM's repair template plasmid for the AAV-S1 locus. The resulting AAV-S1 donor RFP plasmid had the RFP gene under a CMV promoter and a pyromycin's resistance marker under a PGK promoter. Once both plasmids were ready, they were introduced into HEC293 cells using ABM's transfection reagent, DNA fectin. Following transfection, the cells were passaged 10 times to dilute out the episomal donor vector. Following, the resulting HEC293 cells were given 3 to 4 weeks to grow in the presence of pyromycin to select for the cells with successful knock-in of the RFP pyromycin resistance cassette. Cells transfected with both AAV-S1 targeting all-in-one vector and the AAV-S1 donor RFP plasmid were healthy after 4 weeks, and greater than 95% of HEC293 cells were successfully expressing RFP. Control cells that were not transfected with the vectors died after pyromycin treatment. PCR genotyping was carried out to confirm that RFP was successfully knocked in at the AAV-S1 site. In this method, 2 primers were designed. Primer 1, targeting the 5' homology arm upstream of RFP, and Primer 2, targeting a site within the RFP pyromycin resistance cassette. Gene knock-in was confirmed when a PCR product of 1.1 kb was observed, indicating that primers 1 and 2 were both able to bind to the edited genome to enable successful PCR amplification. In the case of a non-successful knock-in or in the presence of wild-type control cells, no PCR products were observed, as primer 2 could not anneal to the target site and enable amplification. We hope this case study has demonstrated how the CRISPR-based gene knock-in technique can make your exploration of gene function amazingly easy and efficient. CRISPR-Cas9 mediated gene knock-in allows for biallelic modifications of some genes to be generated with ease, with phenotypic changes in mice observable in a single generation, an unprecedented time frame. Let ABM's team of CRISPR scientists help you knock in any gene into any cell line you want, all with minimal toxic effects at the AV-S1 or ROSA26 safe harbor sites. Explore ABM's extensive portfolio of customizable CRISPR services and products today, and start knocking in genes, tags, markers, and more. To make it even easier for you, simply describe your desired knock-in, and we'll take care of the rest.